In SATURN, we study the impact of underwater radiated noise on three groups of aquatic animals: marine mammals, migrating fish species, and invertebrates. Scientists of the University of Catalonia in Barcelona investigate the effects of underwater noise on invertebrates by using a special designed lab unit, the Aquavib.
To do so, we have designed and built a specific device, the Aquavib, that consists in two shakers enclosing a transparent acoustic chamber equipped with different sensors to keep the temperature and the level of oxygen in the water and the acoustic pressure and particle motion. We work with three species, Mytilus edilis, the mussels, Nephros norvegicus, the Norwegian lobster, and cuttlefish, Sepia officinalis. In addition, we assess the effects on the larva survival rate and in the success on the hatching of the eggs.
Similar to the Aquavib, but much bigger, is a special technical unit to measure the effects of underwater noise on fishes, the microdome, invented and run by scientists from the University of Leiden.
So here we're standing next to the migrating, a one-of-a-kind swim tunnel where we can control the sound fields that we use as exposures to the migratory fish. First, we will start with migratory stickleback and non-migratory stickleback to test whether we can actually measure migratory behavior. So the migrating is a swim tunnel with an outer basin where we can control the sound fields with underwater speakers, and an inner basin, the swimming part, swim tunnel, where the fish will swim, and because we can tilt it, we can control and create a water velocity gradient. So we can basically, the fish can choose at which water velocity it wants to swim. And so that gives us a measure of swimming motivation for fish. And then we can see how natural soundscapes as well as vessel noise will affect the swimming motivation.
Underwater radiated noise caused by shipping can have a huge impact on marine mammals, their behavior and health status. Therefore, studying the effects of ship noise on this group of species encompasses a large part of the work package 3 within SATURN. Scientists are focused on pilot whales, harbor seals, and harbor porpoises.
The way we are studying the effects of vessel noise in harbor porpoises is similar to the way we do it with harbor seals and pilot whales within SATURN. So the final aim is to try to see if we can compare the results that we obtain. The tags are attached with suction cups to the back of the animal, so we are not directly harming them. The tags contain sensors that tell us about what the animals are doing when we don't see them. They contain pressure sensors that will tell us about the depth at which the animals are. They have accelerometers and magnetometers that will tell us about the movement within the water column. We also have GPS, so we have the geolocation. And finally, we have hydrophones. Hydrophones allow us to record the sounds around the animal, but also the sounds produced by the animal. With this data, we can study if their behavior changes when loud vessels are present. And later on, we can estimate if these changes in behavior could affect the well-being of the animals.
Seals are a common species in the North and Baltic Sea. For tagging seals, it is necessary to catch them at their haul-out sites. These are located on beaches and sandbanks along the coastline, but also in river estuaries like the Elbe River.
These harbor seals live in a major hotspot of anthropogenic activity, including shipping. Around 13,000 ships from all over the world call the port of Hamburg every year. The fact that these seals are exposed to a large number and variety of vessels will help us to understand the seals' response to shipping noise. 8 harbor seals from the Elbe were instrumented with D-tags in the framework of the SATURN project. But this dataset was completed by data from a fieldwork carried out in 2016 and 2017, where 10 adult seals were captured at haul-out sites in the Wadden Sea. The many motion sensors of the D-tag provides us lots of information that allows us to determine how animals respond to vessel noise, as we already do for harbor porpoises. But the fact that the seals are giving a full health check by our vets helps us to combine clinical health parameters with behavioral recordings of the seals. This allows us to investigate if animal behavior is influenced by their health status and if it indirectly influences their behavioral responses to vessel noise.
D-tags are also used for studying pilot whales along the Portuguese coast, around the Canary Islands and the Azores.
The tasks of the University of La Laguna and of the Oceanographic Center of the Canary Islands are to study the response of deep diving whales, in this case pilot whales, to the sound of ship noise, mainly whale watching boats and sounds from ferries that are crossing the area. So with the aid of DTACs, we are going to see how the animals behave in a normal way and how do they react to the sounds of approaching boats. For that, we will also use drones that can see the behavior of the animals from above.
In the open sea, drones are very helpful to study the behavioral effects of shipping.
I use drones to study pilot whales because they are a low-cost, non-invasive technique that allows to obtain high-resolution data on the behavior, the ontogeny, and the group dynamics of these animals.
This is how we are studying the effects of underwater noise on marine fauna as part of the SATURN project.